Good morning, Doug. It's so great to have you. Thanks for making the time. It's really been an honor to have you as the co-director of the Precision Genomics Collaboratory over the past year. And thanks for making time today to talk a little bit about your career and the future of genomic sciences here at Duke. So I'll start first. So Doug, I've been working with you for 2 decades, and looking back on your incredibly illustrious career, can you provide a historical context of what the landscape of human genetics has been at Duke, your career, and kind of put that in context to where we are today?
Sure. You know, when I arrived here in September of 1993, there was a genome project, but there was no genome sequence, there was no genome map. And in fact, one might argue that there really wasn't a genomics or genome science sort of field. And really was driven by, at least in the human genome, by human genetics. And I think that's where Duke has always been reasonably strong in this, originally with Peggy and Jeff Vance, and not just them, but and that whole team which you grew up in. And so when I arrived, that was the world, and we were really focused on doing human genetics and mostly Mendelian disease gene identification. And we were utilizing resources that were slowly trickling in from the Human Genome Project. And as a matter of fact, and I tell this story sometimes to the students, when we identified the genes for cerebral cavernous malformations, what we did with the help of NHGRI was the National Human Genome Research Institute, that is, we built a bacterial artificial chromosome contig over the region on the basis of a lot of clones that we were pulling out to fill in gaps. And then, I don't know if you remember this, but in those days, they didn't, when they sequenced the human genome, they didn't start on one end of chromosome 1 and go like this. They did it in patches, and they did it on the basis of medically relevant regions. And so we nominated that region, and that region got sequenced, and we ended up finding one of the genes through that. And I want to point out where we've got our data, the genomic sequence data was the public domain. We didn't get it, it was just deposited there, and then we pulled it back out and then started doing analysis. So, and even in those days, you had sequence, it was raw HCGT. And you had to like identify genes with all sorts of programs to look for exons and then maybe try to trap exons with exon trapping tricks. So it was the old days. But I do think that that's where Duke had an advantage, because we were, and I think remain, very interested in human genetics and genomics in the context of disease, as you are.
Doug, that's such an amazing story, you know, to thinking about back in a long time ago, where you had to use relatively simple tools, right, to be able to study human genetics. And now you can just get on the internet and look up things. It's really amazing how you have seen that play out at Duke, as well as the really, you know, seminal discoveries. You and I have worked on cardiovascular disease, but also in Alzheimer's and many other diseases. So Doug, what do you see as the future of genetics and genomics at Duke, you know, thinking broadly across human genetics, but also basic science genetics?
I think we have a lot of really good investigators thinking about the functional consequences of sequence variation. I won't even call it mutation anymore, because we've moved so much into complex traits where the variation that you find is already in the population, and it contributes just a little bit of risk. But anyway, you know, whether it's functional consequences at the level of the phenotype, the disease phenotype, or physiology, or especially with Charlie and Tim and Greg Crawford, Tim Reddy and Charlie Gersbach, so I'm speaking of, and Andrew Allen, thinking about variation that what's the consequences for expression, for splicing, for binding, you know, changing chromatin structure. These kinds of things, I think we're prepared to work on and make great advances here. And that's great, because we don't have to have a production facility that beats everyone in the world to make serious advances.
Yeah, that's a great perspective, Doug. I completely agree with you. And the fact that I think Duke has a physically and scientifically integrated health system with the science that we do, right? we're not spread out, we're all in one place. And what I've loved about being at Duke for 20 years is, we can really help our patients, but we can also better understand their diseases in a basic context, right? So being able to delve mechanistically into what we're seeing on the clinical side. And I think what you're talking about with really seminal work that needs to get done in the non-coding or dark genome, and what we can do both ways, helping our patients, but then also using, data from our patients to better understand their diseases in a genetic context. I think that's a great point, Doug.
Right. And that's where I think the collaboratory will come in, is that we need to continue to strengthen those ties and even, build some new ones so that we have more seamless integration of information going back between the clinic and the basic scientists and the translational scientists.
Yeah, I agree. And I'm really looking forward to working with you, Doug, on this, because I think with your deep background in the more mechanistic investigations of human genetics and my role as a physician scientist on the molecular epidemiology side of genetics and translational medicine, I think we have a lot of really exciting things that will come forward in the next few years as we build that landscape of data and information and using it in support of mechanistic investigation, as well as how we take care of our patients. So I'm really thrilled to be able to work with you on that. And I think we have a lot in store over the next few years. So thanks so much for making the time this morning, Doug. It was really great talking to you.
Great seeing you. Take care.
Bye.